Mrs. SHAMALNASAB is biologic researcher from University of Warwick now.
Hello everybody, this is Mehrnaz, and first of all I would like to thank the organizers for inviting me to be here to discuss with you the results that I have done at École Normale Supérieure de Lyon in Dr. Aguilano's lab in collaboration with Dr. Jogger in L'Oréal. And so today I would like to talk about the salicylic acid derivative that extends the lifespan of C. elegans by activating autophagy and also mitochondrial UPR. So here is outline of my today's talk. First, I'm going to talk about the introduction and giving you the background that why we started to work on this project. And then I will talk about C. elegans as a model organisms, and then I will explain about aging pathways more in details, and then I'll show you the results in C. elegans in human cells, and at the end we have the conclusions. So just to give you a background that why we started to work on this project, and actually there are records going back to thousands of years showing the beneficial properties of extracts from willow barks. And these extracts are probably one of the most ancient remedies to reduce fever and also ease aches. So then the scientists were curious to find out what are the molecules that are important in these extracts. So then they found that the main active molecules present in these trees are salicylates. So then, of course, they started to investigate more about the salicylates. So it has been shown that salicylates have beneficial activity on different pathways that are implicated in longevity and lifespan. Actually, they increase the lifespan of different model organisms such as yeast, worms, flies, and also mice. So because this work has been done on worm, I'm going just to talk about how it's happening. So it has been shown that salicylates increase the lifespan of worm by activation of TAF16, which is a FOXO transcription factor and is an insulin pathway, and also by activation of AMPK that is important for sensing the level of the energy in the cells. So having these results led us to investigate another derivative of salicylic acid, which is 5-octanol salicylic acid. So in this derivative, we have an octanol group in a meta position of the acid group. And the interesting part about this derivative is in 2007, it has been shown that it has the lipophilic nature, and also it penetrates slowly in the skin, and it has a skin exfoliating activity. So then we were wondering if there is a positive effect on worms' lifespan. So talking about worms, first I would like to talk and introduce you C. elegans that we used it for our aging as a model organism for the aging studies. C. elegans is a small worm, it's about 1 millimeter, and it has a short life cycle, it's about 3.5 days. And so as soon as they become adult, they give like 300 progenies. And also it has a lifespan of 2 to 3 weeks, which is make it more practical for aging studies, and it's much quicker. Then it has a transparent body, and it's make it as a convenient model for microscopy studies, and we can see all the details of the body of inside the body of C. elegans. It's easy to maintain in the lab, so we feed them with the bacteria on agar plates, and we have large mutants and strain collections, and if we need to keep them for a long time, we can always freeze them at -80, and also genome sequence is known. So having this derivative that I talked earlier, so we wanted to know if it has a positive effect on C. elegans lifespan and half-span. So for lifespan studies, here we are looking at the lifespan of wild-type worms. So first of all, we synchronize the eggs and we let them to grow up until day 1. And then from day 1, we start to count them every other day the dead one and also the alive one. And here we have the wild-type worms exposed to C8SA, and as you can see here in red, they are living around 19% longer than the untreated worms. And also they look healthier for us. So then we study the health span of worms treated with C8SA. And so interestingly, we found that so when worms are exposed to C8SA, they move more intensely. For this experiment, we counted the number of body bends per second, and we found that they move more intensely and also at a later stage of their life. So in another experiment, we also look at protein carbonylation, which is a hallmark of aging. And we could see that when they are exposed with a C8SA, they show less level of protein carbonylation. So by now, we have this derivative that has a positive effect on C. elegans health span and also lifespan. So then the next question is, what are the mechanisms that are involved in the effect of C8SA? So to answer this question, we started to investigate some of the aging pathways. So we know that there are some known aging pathways that by modulation or downregulation of these pathways, we can extend the lifespan in different species. And so they can be like insulin pathway, dietary restriction, or mitochondrial pathway. And also there are some more pathways that here I'm just presenting these three. So we started to investigate the role of insulin pathway to see if it has any role in the effect of C8SA. So in insulin pathway, we have DAF2 as an insulin receptor and also like downstream genes. And so when this pathway is active, it phosphorylates DAF16, which is a transcription factor. And when it's phosphorylated, so it stays in the cytosol. And when, for any reason, this pathway is not active or worms are under stress, and DAF16 doesn't get phosphorylated. So it goes to the nucleus to regulate the expression of the genes that are important for stress resistance or longevity. So then we wanted to know if C8SA has any effect on DAF16 translocation. Is it in the cytoplasm or nucleus? In order to answer this question, we used worms carrying DAF16 GFP, and as you can see here, so DAF16 is everywhere in the cytosol, and when we put them on heat shock, DAF16 goes to the nucleus, so here are the nucleus, and DAF16 is inside, just to regulate the expression of the genes that are important to be resistant to heat shock. And then when we treated them with C8SA, we couldn't see any changes in the translocation of DAF16. So although we had these results, but also it has been shown that even just a little bit amount of DAF16 in the nucleus can be efficient to regulate the target genes. So we look at one of the target genes of DAF16, which is SO3, and it's superoxide dismutase, and it's important for stress resistance, and we could see the level of expression increases. So to confirm this result, we also did some lifespan studies, and here we used DAF16 mutants. So as you can see here, when we exposed them to C8SA, we couldn't see the beneficial effect of C8SA on lifespan anymore. And this suggested us that C8SA extends lifespan of C. elegans through the insulin signaling pathway. So then in next step, we investigated another aging pathway, which is dietary restriction. So dietary restriction is a reduction of food intake without any malnutrition. And for the first time, Mackay in 1935 showed that the reduction of food intake of rodents increases the lifespan. But it's important to know that until 2007, we didn't know about the molecular mechanisms of this effect. So then we were wondering if C8SA also works in dietary restriction. So to answer this question, we did some lifespan studies and we applied different methods to do dietary restriction. So here we have the wild-type worms on ad libitum. AL is ad libitum, and it means that when we just grow the bacteria and we feed our worm with a normal amount of food, which is ad libitum. And dietary restriction is when we diluted the bacteria. So the worms have less bacteria, and as you can see that they are living longer. And then the third condition is bacterial deprivation, and it's when worms are adult, we put them on plates without any food. And it's very interesting because C. elegans is one of the rare model organism that can stand the complete bacterial deprivation. So there is no food until the last day, and you can see that they're living much longer. So then when we exposed the worms on C8SA, we couldn't see any effect of C8SA anymore, and that also suggested us that C8SA might work in dietary restriction pathway. So as I said, until 2007, we were not sure about the molecular mechanisms, and then there were some genes and mediators that we found that these genes and mediators are responsible for the effect of dietary restriction response. And so we tested some of them, and today I'm just going to present 2 of these genes. So the first one is AMPK. As I showed at the beginning, it has been shown that salicylates increase the lifespan of worms by activation of AMPK. And we wanted to know also if this is the same case for our derivative. And we found that when we used the AAK2 mutant, so in AAK2 mutant, we don't have the activation of AMPK anymore. And we could see that the effect of C8SA on lifespan extension is missing here. So this suggested us that C8SA increased the lifespan of worms by activation of AMPK, or in another word, activation of AMPK is important for the effect of C8SA on lifespan. Then in next step, we moved on to TOR signaling and autophagy. To check TOR signaling, we looked at one of the targets of TOR, which is DRR2. It's a eukaryotic translation initiation factor, and we could see that when we exposed the worms to C8SA, we could detect a significant reduction of the expression of these genes. So then this result suggested us that TOR is inhibited when we move the worms to C8SA. So then if TOR is inhibited, so then we were expecting to have more level of autophagy, and actually that was the case. So to do this experiment, we used transgenic worms that they were carrying LGG1 GFP, which is an autophagy readout. And as you can see, there are some cells here that I'm showing them here. So we call them SIM cells. And then we can count the number of the foci that they are the autophagies or accumulation of the LGG1 GFP. And as you can see here, so as a positive control, we have dietary restriction that increases autophagy. And also when we expose the worms to C8SA, we could see that the level of autophagy is increasing. And then for the negative control, we have BEC1 RNA is one of the autophagy genes, and as soon as we don't have it, so the level of autophagy decreases. So talking about BEC1, and also we wanted to confirm these results and show that the autophagy genes are important for the effect of C8SA. So then I use another mutant, which is autophagy mutant, and we could see that we don't see the effect of C8SA anymore. So we can say that C8SA activates various processes usually linked to dietary restriction. So this is the last aging pathway that we also tested. It's mitochondrial pathway. So we know that modulation or downregulation of the subunits of mitochondrial complexes increases lifespan and also triggers mitochondrial UPR, which is unfolded protein response, and is important for the survival of the cell. And that also increases lifespan. So in order to see if we have C8SA is functioning through mitochondrial pathway. We use one of the mitochondrial mutant, and we could see that the positive effect of C8SA is not there anymore, and that suggested us that C8SA is also working in mitochondrial pathway. So then we wanted to know if the mitochondrial UPR is important for this effect. And here we have the mutant that are defective in mitochondrial UPR, and we could see that the positive effect of our compound was not there, and we don't see the further longevity extension, and we could conclude that activation of mitochondrial UPR is important for the lifespan extension of effect of C8SA on C. elegans. So by now, we know that C8SA is working in insulin pathway, dietary restriction, and also mitochondrial pathway. And here are the mediators that are important for the effect of C8SA. So we know that it increases lifespan through activation of mitochondrial UPR, autophagy, DAF16 transcription factor, and also by activation of AMPK. So now, what about skin? Because we know that worms, they don't have skin and they just have cuticles. But the good news is, when we started to do this project and when we had this nice result on C. elegans, we were hoping to show the conservation of the role of C8SA in higher model organism, or let's say more complicated, such as cells. And actually that was the case. So then we were collaborating with a lab in the University of McGill, and we found that C8SA activity is conserved also in normal human keratinocytes. So first of all, we measured TOR activity, and by measuring the phosphorylation of RRPS6, and we could see that treating the cells with 30 macromolar of C8SA decreases the level of phosphorylation. That also confirmed the results from C. elegans. So by having less TOR, we were expecting to have more level of autophagy, and that was the case. And we could see that C8SA induces autophagy in a dose-dependent manner in cells. So also, we showed that AMPK is active in cells when they are treated with our compound, which was the same case in C. elegans, and we did it by measuring the phosphorylation of the PACC, and we see that after 3 days of treatment, the level of phosphorylation of the target is increased. So just to conclude, we can say that C8SA extends C. elegans lifespan and health span, and also it increases longevity of worms by activating autophagy and mitochondrial UPR. And also it increases autophagy in human keratinocytes. And as I showed you before, in 2007, so we found that this derivative has exfoliating activity. And so we can say that also it might have some anti-aging benefits on the skin, because there are some other results that I didn't show you here, but we showed that it's also important for the differentiation and also mitochondrial biogenesis. So if you're interested more in the results, we can always talk, or here is the paper, and that is just published in Aging Cell this September. And at the end, I would like to thank Dr. Aguilanius at ONS, Dr. Jagger at Paris, and also L'Oreal, and also our collaborators in University of McGill. And thank you very much for your attention, and I'll be happy to have any questions.
Thank you very much, Mrs. SHAMALNASAB, for this very nice presentation. Is there any question in the
Thank you.
assistance? Microphone.
Merci. Thank you very much. Actually, two questions. One is, Your derivative, does this also have the COX-1, COX-2 inhibitory activity of salicylic acid? And if did you look at that? And no. The second question is, because that might be even upstream, from what you observe. The second question is, did you compare the potency of that derivative to known TOR inhibitors like rapamycin or the homologous substances?
So for the first question, sorry, no, we didn't check that one. And for the second question, if I was understood very well, if we did it with a TOR? No, right. Rapamycin?
Right. Since you're inhibiting the TOR pathway, a control that you could have run side by side would be to use, rapamycin or rapalox to see the potency of how good your molecule is in comparison to the to other TOR inhibitors.
Actually, yeah. Here we have rapamycin, and then we can see that even the effect was just, but also for sure it depends on the concentration. But the effect was more significant by using the R derivative.
Beautiful, thank you.
Yeah.
Question, Lionel Breton.
Thank you for your talk, Mehrnaz. So just to answer to your question about COX-1 and COX, Professor Grilari, just to answer to your question, the compound have no effect on any kind of COX activities. That's why it's of interest for L'Oréal Corporate. Just to answer to your question. Mehrnaz, just to go back to the classical fields of C. elegans and all the people working on these fields. So, do you know if, because you are more involved in these topics than we can do,
yeah.
Do you know if Calico Corporate, which is for anyone the global anti-aging firms created from scratch by Google 2 years ago, do you know if Calico Corporate is working on the same target, for example, and mentions that in classical communication or something like that? Why I ask this kind of question, because I see my colleagues, they are laughing just on my left, is so, because the main expert of the world working on C. elegans is a close, very close number of people and experts all over the world, and Calico probably is one of the main competitors to propose something in the future to prolong healthy span of human. And the question is this kind of topic is of target is of interest for us, or if it's not so diffuse and it's for the moment extremely secret by the company?
It's a very good question, because I would like to know too, because whatever Calico is doing is confidential. And okay, no, it's okay. So we had a postdoc in our lab that moved to Calico, and now it's just like 5 years that he's working there. But whenever we ask him any question, he won't answer it because everything is confidential. But for sure, we know that they work a lot on C. elegans and also in cells. But I think they just work on C. elegans because it's much faster, and most of the genes are conserved in human cells and also in other model organisms. So that's why they started to work on C. elegans. But if they're interested in this one, I have no clue. I can ask my friend if you want. Yeah, I told you, maybe at the beginning you didn't hear it. You're welcome.
Okay. I have a question about the skin part of your talk, sorry. Do you know if the exfoliating action of the C8-AC could be linked to the modulation of keratinocyte differentiation by autophagy in the epidermis? It was studied by your collaborators?
as I said, it's done with our collaborators, but we know that there is also changes in the, so there were some markers, like differentiation marker, were also upregulated. And we know that because when AMPK is activated, and because we showed that AMPK is activated, so then it activates the target genes. And it's important also for activation of some of the differentiation genes, which they are there.
Thank you.
Questions there?
Thank you very much for the interesting talk. I have a translational question as well. So at least for your in vitro assays, you use quite high doses of 30 to 100 micromolar. So, and this is quite high doses, speaking from a pharmacological perspective. Can you comment on that?
I mean, you're talking about this experiment, yes?
Yeah, but the doses for rapamycin are 100 nanomolar, and for your compound you have to use 100 micromolar. So the question is, would this be applicable to oral treatment, or would you need such high doses that this is not possible as a treatment?
To be honest, I wouldn't, I don't know this one, yeah. Because, I mean, I was responsible for the works on C. elegans.
Thanks.
But, I mean, in C. elegans, we did a dose dependent, and we found that the best concentration was 100 micromolar. Yes.
No more questions?
I was wondering if there is any effect of your compound on ROS production, and if ROS production has any role in the extension of lifespan and etc.
We didn't check if there is any effect on ROS production, but what we checked is we had less level of protein carbonylation, which showing that we have less level of oxidative stress. So if I answer your questions.
Okay.
Like it can be like indirectly showing that we might have less level of ROS production.
Last question, after we have to continue.
It's a comment, it's not a question. Just to come back to the dose with the rapamycin. Rapamycin is a direct inhibitor of mTOR, and the effect of this compound is indirect. So I think that would explain that you have you need much higher dose because it's an indirect effect of AMPK activation and so on. That was just a comment to answer this question. Thanks.
Okay, thank you. Thank you very much, Mrs. SHAMALNASAB.